So I'm very excited to introduce our speaker today. This is Matt Kustra. He did his undergrad at University of Virginia with highest distinctions. And then we did our PhDs together at UC Santa Cruz. We were in the same cohort where he worked with Dr. Suzanne Alonso on awesome sexual selection stuff. Maybe we'll hear some about the oscillated grasses today. And he is now a Miller postdoc fellow with Chris Martin at UC Berkeley. He's had all kinds of awesome awards from things like NSF and ARCS and AMNAT for research, etc. Lots of great publications in a bajillion journals, including some you may know of like AMNAT and Evolution and Functional Ecology. And he's also just a really great dude. And I hope you guys, if you're around, join us for beers at Dunlow Local after this. We'll be hanging out, having a couple beers around 5:30. And with that, I'll hand it over to Matt.
Well, thank you for that lovely introduction. I'm really glad to be here this afternoon to talk a little bit about my research. So when we typically think of sexual selection, we think of traits like this: bright ornaments, elaborate behaviors, ornaments, and this is really only half the story. In a lot of species, females mate with more than one male, and this can generate postmating sexual selection, or selection on traits that aid in fertilization success, such as sperm. And so when we typically think of sperm, we think of a small tadpole-shaped like cell like that. However, when you look across the tree of life, sperm are actually the most diverse cell type. And so pictured here are sperm from different animals from across the animal kingdom. And although this isn't to scale, they also vary largely in size. So the smallest is a parasitic wasp that's only a few micrometers in length, to the largest, which belongs to a fruit fly. So this is a single sperm coiled in a ball, because it's about 6 centimeters long, which is 20 times larger than the male itself that actually produced it. And so you have this amazing diversity. Once again, you usually think of a tadpole-like thing. A lot of sperm have evolved to lose the flagellum altogether and actually climb like an amoeba. And you also have other evolved apparatuses like these helical structures have evolved at independent times. And so as well, I'm really interested in this diversity that we see both through pre-mating sexual selection as well as post-mating sexual selection. But a lot of my research is focused on post-mating sexual selection. I'm particularly interested in studying the ecology and evolution of reproductive interactions that's driving this diversity. And so 3 main questions that my research program seeks to address is: how's the environment influencing these reproductive interactions? So you can think of previous work that I won't talk about today is looking at how microbes might influence reproductive interactions. Today I'll be talking about social environment and temperature. But how does these interactions influence the evolution of life history traits and speciation? And then finally, something that I've been diving more into in my postdoc has been looking at sort of the genetics underlying speciation and these reproductive trait evolutions. And to do this, I use a variety of methods. So both these theoretical, often individual-based simulations, but also some analytical models, field work, experimental work, and as I said, during my postdoc, I've been stepping into genomics. And so for today's talk, I'll first focus on some of my PhD work on the oscillated RAS, looking at how social environment and temperature influence reproductive interactions. Then I'll talk about some theory I've been working on, looking at the role of post-mating sexual selection in maintaining reproductive isolation. So thinking of post-mating pre-reproductive or pre-zygotic reproductive barriers. And finally, we'll be talking about work I've been looking at genetics underlying this adaptive radiation of pupfish in Chichikov in the Yucatan. And so the first section of the talk, I'll be talking about how social environment and mating strategy influences the timing of mating. And so work of this is on the oscillated RAS or Symphonis ocellatus, and it's in Corsica. And so unfortunately I have to do field work in this lovely field station, but it's convenient because we can both do experiments bringing up at the lab, but we also, the fish build their nest on pretty much on the rocky reefs that you can see right there. And so what we do is suit up, dive on in, and then we swim to find these nests. And so they'll build their nests sort of in those little rocky reef areas right there. And so that we just found a nest there, and zooming in, what you might see is these nesting males, they build this algae nest. And so nesting males, they make this nest, they court females, and they provide all the obligate parental care. And so if there's no nesting male present, all the eggs, there's zero survival. And then you also have this tactic, sneakers, they're much smaller, and they'll just wait for the nesting male and female to spawn, and then they'll dart in and try to fertilize the eggs. And this happens in a lot of different systems, which makes this system a little more unique. We have this third type, a satellite male, which is about intermediate in size. And so they pair up with the nesting male, chase away sneakers, court females, but then they also sneak. So there's this cooperation conflict dynamic. And previous work, just to let everyone know, has shown that this isn't like a case of kin selection. So the satellite male isn't more related to the nesting male than any other random fish. And so a bit what we know about the life history of these fish. So if you're a first year with fast growth, you'll be a satellite male in your first year. And then second year, you'll be a nesting male. If you have slow growth, you'll be a sneaker male in your first year, and then a satellite in your second year. And so what do these nest dynamics look like? So pictured here is a nest. And so the female is actually already in the nest. It's about to spawn with the nesting male. Then you have a satellite and sneakers. And so the nesting male, they just spawned with the female. And you can see that they're already oriented about to sneak. Satellite went in, released sperm, trying to fertilize eggs. And then you have sneakers slightly behind looking at that. And so just watching that in real time. So this is all happening really fast. And we know in a lot of systems that actually just small fertilization happens so fast that just small delays influence probability of fertilization success. And so what I was particularly interested in is that a potential benefit of being a satellite? As you can see in the video, the satellite was typically allowed closer to the nest, are able to get to the nest faster relative to the sneaker male. And also, we have quite a bit of variation in like the social environment during the sneak mating. So there's 2 sneakers there and a satellite. The most I've seen in the wild has been like 20 sneakers all around the nest to see this like crazy amount of variation. And so look at this, did a bunch of 10-minute live behavioral observations with video recording. And this is across multiple years and different nests. We're analyzing these videos to get specific time differences. So from when the nesting male spawns to satellite or sneaker. And because this is very hierarchical, I used a Bayesian generalized linear mixed model effect. So I won't be presenting p-values, but rather posterior distributions. And so I looked at both the male tactic, but then also the social environment. And so what I did was sort of bend it to different categories. So you might have a sneaker or satellite sneaking alone. That happens quite often. So very little competition from the perspective of the sneaker. You might have the satellite and sneakers both sneaking at the same time, or you might have many sneakers and a satellite sneaking at the same time. We typically only see one satellite at a nest at a time. And so just to walk through the graphs I'll show, satellite male is going to be in blue, yellow is sneaker, and plotting the posterior distributions with the 95% credible intervals given by those dashed lines. So this is actually what we saw with just when there's a single sneaker or a single satellite. So satellite is generally getting there faster, but they still have overlapping credible intervals. In that scenario, when there's both a satellite and a sneaker, sneakers are actually getting there a little bit slower, and we do even a bigger effect of satellite getting there faster in that competitive scenario. When it gets really crazy, when you have a bunch of them, sneakers are actually, on average, individual sneakers getting there faster than the other scenarios that are much closer in time. So that might be benefiting potentially from these sort of chaotic scenarios. So just to sum up this section, so you've got shorter time delays in spawning is likely the benefit of cooperation from the satellite male standpoint. Sneakers are maybe benefiting from busier nests. And I'm happy to talk about this outside things, but a lot of these sort of dynamics of these like local social environments is sort of not incorporated in a lot of sperm competition theory. And this is actually where there is a breakdown between like some of the theoretical predictions and the empirical aspects. I'm interested in trying to tie that in. So this is all one aspect of social environment, but then afterward, there's sperm competition, and that's what I'm particularly interested in. And while I was conducting some of these surveys, there's actually some pretty bad heat waves in the Mediterranean. It's really got me interested in how temperature might be influencing some of these sperm interactions, sperm-female interactions. And Indeed, the Mediterranean is a global change hotspot where it's warming about 20% faster than global average, and there's been a lot of heat waves. So just to orient everyone, the field station is right over there. And one of the nice things about working in a field station is we actually have some pretty good long-term data. So what's actually happening in this specific place that we're studying, going back to the 80s. And so shown here is just like across the breeding season. So the breeding season's from early May to early July. And this is just from across the 40-year data set. The median is the white line, and then just the variation. And so I'm really interested in temperatures vary both within and across seasons. So this is just a lot of variation happening within the season. So that's interesting and how that might be influencing these reproductive interactions. But as I mentioned before, we also have well, that jumped like crazy. Okay, anyways, both, sorry. And then across the seasons, if you look at just maximum temperature from the field data, we see that there's a clear increasing trend from the 80s up to now. And actually, just this past year was one of the warmer years that we've experienced. And so I wanted to choose test temperatures that were relevant, particularly 16, 22, sort of representing beginning and end in red. And I do want to highlight that although 16 C is at the earlier part, it's skewed that they're mostly mating actually in that first week of May, first 2 weeks of May, even though they'll breed across there. So that's clearly closer to what they're actually experiencing. And the interaction that I'm particularly interested in is looking at sperm ovarian fluid interactions. So these fish are external fertilizers. And so when females lay eggs, they have this fluid coating, and we generally call that ovarian fluid in the fish world. And it plays all sorts of important aspects. So it helps sperm find eggs, it helps sperm live longer, but it can also influence fertilization. So in this cartoonish example, you see ovarian fluid is sort of like filtering out some of those blue sperm from a different male. However, if you remove ovarian fluid, it's about 50-50. And while this is a cartoonish example, ovarian fluid it influences fertilization dynamics in this system. And so previous work done by my advisor to show that it influences the importance of sperm number. And so if you run like fertilization trials without ovarian fluid, pretty much get a perfect correlation between number of sperm and paternity, which is what you expect. However, with ovarian fluid, sperm number no longer is a big predictor. It's actually sperm velocity is about 40% of the prediction predicts 40% of the variation in paternity. And what this actually does is essentially bias it towards nesting males. And so we know that actually in the field, like in a natural spawning, sneakers release about 4 times as much sperm as nesting males. However, nesting males generally have faster initial sperm velocity. And so it's really sort of trying to counteract that, it's counteracting that sneaker numeric advantage. And for this experiment, I'm mostly focusing on sneaker males and nesting males. That helps make it more logistically tractable, but then also we know that there are different life history pathways. And so for this experiment, took a female, you collect and dilute ovarian fluid with seawater and incubating at those 3 different test temperatures that I was talking about, 16, 22, 28. And then we take the nested male or sneaker male, and we're recording their sperm velocity in these different files. And so one of the nice things is that I had a temperature controlled stage, and so actually recording it at the temperature that I wanted to. And also to control for individual variability, each male is having their sperm across all 6 replicates. And we did about 38 right now. And so what this looks like. So one of the nice things, so have a video tracker and you can see how fast the sperm are swimming at these different, with or without ovarian fluid, different temperatures. Luckily, I don't have to track that myself. And so we have automated trackers that do that for us, and they can actually record all sorts of different metrics. And so I'll be talking about the curvilinear velocity, because as you can notice, a lot of the paths are sort of circular. And so I found a 3-way interaction. Woo! And so I'm going to walk slowly through these results. And so it depended both on whether it was seawater, ovarian fluid, the male type, and temperature. And so just in seawater, we see that as temperature increases, especially from 16 to 28, there's an increase in sperm velocity. And so anecdotally, sperm velocity just increased at warmer temperatures until roughly like around 33. That's when it died down. But that wasn't really as relevant to what they would be experiencing. So at these turns, it doesn't seem like it's sperm are losing their mobility. In ovarian fluid, there's actually no effective temperature for nesting males. The effective temperature goes completely away. However, for sneaker males, we still see a positive effect of temperature. And so just looking at the effect of ovarian fluid, so we do expect that ovarian fluid should be increasing sperm velocity. That's what previous work in the system has found, and it's found in like most other systems. And we do see that at this coldest temperature, 16 C, which is closest to when peak spawning is for both nesting males and sneakers. However, at 22, that effect goes away for nesting males, but it continues for sneakers. It's a little bit lower. And then at 28 degrees, there's no positive influence of ovarian fluid. So it's really only having that influence at the cooler temperatures and only at 16 C for nesting males. And as someone interested in sexual selection, this is just looking at those individual interactions, but what does that mean in terms of predicted fertilization success? And so we can look at the difference in sperm velocity between sneakers and nesting males. And so just to orient everyone on this graph, if you think of the positive, that means nesting male sperm are swimming faster. On the gray, sneaker male sperm are swimming faster. And so you can think of a nesting male wins, sneaker wins. And at 16C, but we do see that that's what we expect. That's what previous work has shown, that nesting males are sperm are swimming faster. However, when we get to 22, it's now sneakers, and same with 28. One of the nice things is that we know, although I wasn't able to actually run the actual fertilization experiments, we do know how sperm velocity should influence fertilization success. And so if you sort of plug in that velocity into other models that we know relating to that, we can sort of get an idea of reduction in nesting and returning. And so going from 16 to 22, we see about like roughly on average 8% predicted reduction in paternity. Same thing if you compare 16 to 28. But 22 to 28, there's no real difference. And you sort of saw that velocity. So it's really that 16 to 22, which is also interesting because 22 is pretty much right as reproduction is ending. So we're sort of on that edge, if you will. And so just to summarize, climate change may be undermining cryptic female choice of the system. There's still a lot of work that I'm really interested in pursuing to do this, but we're seeing this positive impact only happening at these cooler temperatures. Warmer temperatures potentially bias paternity towards those unpreferred sneaker males. And we do know that sneaker males sort of represent lower quality, so we're really interested in what this might mean for population dynamics, but it's all very speculative, and really interested in digging into that a bit more. Okay, so talked a bit about the RASes, and now I'll be talking about postmating sexual selection in the context of speciation, some of the theory that I've been working on in my postdoc. And so most of these models I'll be presenting are individual-based models or simulations, looking at the role of postmating sexual selection and reproductive isolation. And so just to go back to sort of grounding, what really got me in this framework was thinking of like this amazing diversity that we see across species of sperm, thinking of how that might relate to speciation. And although I'm just showing the male component, when in like internal fertilizers at least, the female reproductive tract is also really diverse. So there is evidence of like coevolution, co-divergence between those things, sperm length and female reproductive tract. The one I thought of how this might be happening is through non-directional cryptic female choice, or essentially you might imagine that there's like a G by G interaction. So if male one sires 20% offspring, male two sires 80% of offspring when mating with this female, we might, if we just took a male mediate bias lens, we might say, okay, male two just has better sperm. But what's been found often is that there's this interaction. So if you take a different female, same males, we see that pattern's reversed or just not as strong. And so sort of the outcome becomes is dependent on both the male and female type. And so I'm really interested in modeling how that might relate to some of this co-divergence. And to do that, you can think of each female as creating an optimum, and then think of different strengths of selection. So it's a Gaussian function, but stronger selection means like closer you are to the optimum, the better you are. And then sort of strength of selection means how far away you can get without shuffling fertilization context. And so one female might be there, and another female might be there. And so I'll be modeling all like quantitative traits in this, so not population genetics. And so one of the first things was just like, do you get those co-divergence of postmating traits? Can you get like those statistical associations that we think of like in fishery and runaway sexual selection? Under what conditions? And so as someone that studies sperm competition, one of the main things that we're interested in is like the rate of multiple mating. And so just showing you some results of these simulations. And so here's like a perfect correlation. That's just like starting point and just different replications of the simulations. So to quickly go to that line and then just through genetic drift, they're sort of drifting along that line of equilibrium, if you will. And so I'm showing you this just to highlight that this is actually occurring even when females are only mating once with like multiple males a quarter of the time, and in that weak selection scenario. So it's not actually that strong. You can just still get these effects over generation time. And what I was particularly interested in is we see that there's been this co-divergence, like in these isolated, you can think of individual simulations as isolated populations. You see that co-divergence, but how effective is that actually at maintaining reproductive isolation in like a secondary contact scenario? And so, for example, you might just take the ends of news and simulate what happens. So there's different scenarios that we might imagine. You can imagine where there's only been preference divergence, but no ecological divergence. By preference, so I've already mentioned one example, like sperm size and female reproductive tract length. That's actually been empirically shown to be like one of these dynamics. You also might think of like pH has a big influence. So it could be, the pH of the ovarian fluid or reproductive tract allowing for optimal function of the sperm, for example. So think of those quantitative traits. Alternatively, you might have something where there's been ecological divergence, but no actual preference divergence. What's happening in that scenario? Or sort of the best case scenario when you're thinking of speciation, you have both ecological divergence as well as divergence in this. And so in these simulations, I don't have, there's no pre-mating sexual selection. So mating's all random. Just keep that in mind. So only like sexual selections occurring after mating. And while I have this in mind of like animals, this could apply to plants, thinking of like pollen pistil interactions as post-mating prezygotic reproductive barriers. I think there's a lot of similarities and could be related to that as well. And so just in that first scenario where there's no ecological divergence, just the sexual selection divergence. And right now I'm also just plotting, my metric of reproductive isolation is divergence in neutral markers. So not actually the traits under selection, just randomly neutral. And it's this very simplified model. So none of the different alleles are physically linked. So it's all recombining. And so you do get maintenance, interestingly, so on the x-axis, it's sort of the rate or degree of the multiple mating. And so you might expect that higher multiple mating is stronger selection, better resistance, but that's actually not the case. We see it's intermediate. I'll get into why. And then on the y-axis is the migration rate. So how often are migrants coming from one population to the other? And then sort of the darker the blue, more reproductive isolation is maintained. And then there's weak, moderate, strong choices. And so what's going on here? And so essentially, so this is actually from an analytical model, just to give insight that's more controlled, not in the simulations, is that essentially there's actually intermediate in terms of just thinking of the probability of successful migrant male, or I'll just say a male from population B. So if there's no multiple mating up there, essentially everything's random. So random mating, random fertilization success, it's determined just by the proportion of males from population B, which is roughly 0.0, which is like 1%. However, as you increase the rate of multiple mating, perfect female choice is acting to select against those other males. And so the successful mating from population B is actually going down. And while this is dependent on the frequency, essentially, if there's a lot of multiple mating as it approaches infinity, essentially what's happening is that you get the reverse happening where the migrant females are also able to select and more likely to encounter other males from population B. And so you maintain those alleles in the population, essentially. And that's essentially what's going on to generate that intermediate effect and having to talk about that in more detail later. If we just have ecological divergence and no preference divergence, that actually does quite a bit for this model. And so, interestingly, we do see quite a bit of sexual selection happening. And so if you look to, one of the interesting things is if it's just ecological divergence, multiple mating shouldn't have that big of an influence. But we do see once migration rate gets to 4%, that we're seeing that something's going on there. And so what's happening? And so essentially what's happening is that cryptic preference divergence is actually evolving through reinforcement in this scenario. And that actually is like essential for reproductive isolation in terms of like looking at neutral markers there. We also see, I just want to highlight, we also see it down here that this divergence in cryptic female preferences are evolving as well. And so just to show like when it's not high enough, high enough, you do actually get the evolution of cryptic preference divergences in some populations pretty rapidly. And essentially once that divergence happens, gene flow sort of gets restricted and so maintains some neutral divergence between the different populations once sufficient evolved. And so going over a bit what's going on here is that in some populations, as you can see from the previous graph, there's quite a bit of variation. And so there is a lot of stochasticity to this process. But just zooming in on some of these replicate populations, you see that the correlation between the male trait and cryptic preference trait and the ecological trait, they can evolve oftentimes. And in those populations, some of those green and purple lines, you actually start getting selection for divergent cryptic preferences acting on in those populations. And once you get selection on divergent cryptic preferences, you do get the evolution of divergent cryptic preferences. And in those populations are the ones where you see gene flow stopping between those two populations. And some mutual variation is fixed between the different populations. If you have the best case scenario, we see that pretty much you're able to maintain pretty strong reproductive isolation across most scenarios. And so just to conclude with the section, we see codivergence in these like post-mating reproductive traits occur even with low multiple mating and weak preferences. Under certain scenarios, cryptic female choice on its load may be effective maintaining reproductive isolation, but relatively limited. If you throw in ecological divergence, even weak cryptic female choice can help contribute to reproductive isolation. Finally, you can get these reinforcement in these cryptic preferences that can help maintain reproductive isolation. And so that concludes that section. And so now I'm going to be focused on some of my more recent work that I've started as a postdoc, looking at sort of genomics underlying adaptive radiation of pupfish in the Yucatan. And so the star of this section are printed on pupfish, specifically the radiation in Lake Chichikanab. And so if we zoom in on Lake Chichikanab in the Yucatan, it's a relatively small skinny lake, and this is sort of what it looks like. And here is us sampling with a bunch of field teams and professors from UNAM. And in this lake, we have some specialists, three different pupfish species. So we have Beltrani, which is sort of a generalist, which is like most pupfish. We just sort of eat detritivores. They just sort of eat detritus. Then we have Labiosis, which is a bivalve specialist, and Simus, which is a zooplanktivore. And you can sort of see it here, but showing some CT scans is they've diverged drastically in their jaw morphology, especially Simus. So I just want to highlight how angled the jaws become relative to the other fish. We also see pretty big, strong divergence in teeth morphology. That's one of the biggest divergence for Simus. Which makes the system really nice and interesting is that we have another radiation that occurred roughly around the same time point. And so in San Salvador Island. And so we know a decent amount about what's happening in that radiation. So we can sort of try to ask questions about the degree of parallelism in these different radiations. So we're looking at sources of this adaptive variation, sort of the genetic architecture of this variation, trying to think of the stages of selection and the relative role of sexual selection. And so just to compare, we'll be going through these different lists of what's going on in different populations. So like I said, they're formed roughly around the same year. So these are all really young incipient radiation, so 8 to 15,000 years old. And so in San Salvador Island, you have, once again, you have a generalist, you have a muscivore, and you have a scalator. And it's not, though it's not the same, you do see similar relative like ecological divergence. And we know that in San Salvador Island that these are genetically distinct. In Chichikanab, we didn't used to know what they were, it's mostly morphology. And so the first thing was just looking at sort of, do we cluster out nicely genetically? And so shown here is a sampling site of artifrons, which is like the sister to the radiation. It's not like ancestral population of artifrons colonized that way. And so just looking at principal component analysis of whole genome sequencing, we do see that they cluster out nicely, especially PC1 is mostly dividing C. artifrons from, which is the sister species from the ones in the lake. And within the lake, we get nice clustering, especially for thymus. For San Salvador Island, we know that introgression played a big role from different islands in the Caribbean, as well as from mainland America. And so I was curious to see if we're seeing strong patterns of introgression in Lake Chichikanab as well. And essentially, we did not. So this is plotting full F branch statistics. And so bright red would be a lot of introgression. And so these are all the sort of like the outgroup populations that we do see introgression coming in from for San Salvador Island. We're using actually some of the exact same samples, pretty much all white there. We would see a bunch of red happening in that if we saw that. So pretty limited introgression in this case. And so I was interested in what are the potential barriers for introgression. And so there are both sort of islands. This is landlocked, of course, but there are like underwater crabs, and like the whole Yucatan is pretty porous too. But one of the also things that makes Yucatan or Lake Chichikanab pretty unique is that it's saturated in gypsum and sulfite, which is pretty toxic to a lot of fish. And these are also external fertilizers. So they're releasing their gametes into the open water. And calcium regulation and calcium has a huge influence on sperm. And so I was interested in looking at sort of local adaptation of sperm to that environment. And so we went out in the field. I brought a microscope down into the hotel, collected some sperm, and we had collaborators from one of the Yunnan research stations bring C. artifrons nearby, and we could look at this. So here's some ocean water, which is where C. artifrons is from. It's not great, but it's still swimming. This is the Chichikanab water. That is moving, so this is a real video. Well, this is a cartoonish example. At least out in the field, we see that classic crisscross that you expect for local adaptation. And I've repeated this experiment in a more controlled setting. So I have F1s that have all been commonly raised, and we see the same pattern. So it seems like they're at least is most likely genetic component. And I also have F2 hybrids that I'm trying to do some QTL mapping of this right now, but that takes a while, so I don't have that right now. Anyway, so just to highlight that, we see limited introgression in Chichikanab, but a lot of introgression in San Salvador Island. And most of the candidate variants we find in San Salvador, like the variants that we think are under selective sweeps are non-coding. And so we wanted to see if that's the same, which makes sense due to how young this radiation is. And so first like filter was just looking at SNPs that are fixed between these species or nearly fixed. And so for Cybus, we identified some, but Beltrane and Labiosis, they don't really have anything unique at that threshold. But so for the purpose of today's talk, I'll just be mostly focusing on Cybus. And so that's the first threshold. Next thing I did was try to look at selective sweeps using population branch statistics, and sort of overlapping those two, we get about 661 SNPs, and then 425 are within 15 to 20 kb from your gene. And just highlighting where we find those, they're scattered throughout the genome, so it's not like it's concentrated on a single chromosome. And I'll just point out that all evidence points to they don't really have diminutive sex chromosomes anyways in this system. And then when we look where they are, like the ones that are near a gene, where they are, most of them are in flanking regions, quite a bit in intronic regions, but we do see some non-synonymous and synonymous mutations. And so that's something that we're seeing in both these radiations, that most of these are non-coding. One of the interesting things, at least in Simus, about 2% of those variants are likely de novo, so not found anywhere in our wide sampling across the Caribbean pupfishes. And so what's going on in this place for our Simus? We find that about 30% of those variants, using the same data sets, we can't find anywhere else, which suggests they're de novo. Most of it's standing variation, but at very low frequencies. And just sort of comparing that pie chart for the scale leader Simus, we see, once again, introgression. A lot of those alleles were in introgression windows. We don't see that in Simus. The next thing I wanted to do was look at trying to see if we can time these selective sweeps. And so taking those genes, seeing those variants, when likely were they going under selection? And so this is sort of just estimated of like the age of these selective sweeps across the different genes. I was interested if something about the category can tell us. And so what we're finding is that the de novo genes are sweeping most recently, those de novo variants. We see a strong pattern there. We also see a similar pattern looking at non-synonymous mutations, but not as strong, partially to the lower power. We don't have as many non-synonymous mutations. And so what might be explaining this pattern, we might expect this with de novo mutation. If they are de novo, then there's sort of a time lag for a de novo mutation to actually arise, but also a more adaptive approach could be that they're de novo for a reason that in a normal genetic background could be deleterious. But interested to expand on that a bit more. And we see a similar pattern actually in San Silva Island. So these de novo variants are sweeping last in that radiation as well. And finally, just looking at sexual selection, many of the pigmentation, there's a lot of genes that are been characterized as related to pigment, and you can sort of see that with these examples, but we don't really see any of that going on in this radiation. We actually did find several spermatogenesis genes popping up in this radiation, which, you know, as you saw the beginning of this talk, or the rest of this talk, this sort of excited me. It was lucky. And so we actually looked at some of, looked at the sperm in these species. And this is mostly led by a great undergrad, Fifi. And so just looking at sperm, we do see that actually quite big difference between Beltrani and Simuses. They have like non-overlapping distributions. And of course, the sample size is limited, in part due to Simus is actually extinct in the wild. And so we have very limited samples now, unfortunately. Another thing is we also, when looking at the genes, although there have been a lot of similar craniofacial divergence, there's actually very minimal overlap, and so the same genes are not involved in both these radiations, essentially. But we do see many visual and craniofacial things popping up, which is what we expect. So Simus is a planktivore, it's really changed its ecological niche. And so it makes sense that vision might be important. And so one of the things that we wanted to look at is there visual acuity differences between these different species? And this was led by undergrad Dylan. And so we use this optomotor response. So you put a fish in front of a black and white spinny thing, you spin it, and then if they have a strong optomotor response, they'll try to track the black or white that's like spinning around. What this looks like is like, so that fish has a very strong optomotor response. So it's moving around, and you can do various controls, which we do, like spinning without anything, and then there's no response. So it's really responding to those black and white lines. And Beltrani pretty much has like no response. They'll just like sit still throughout it. Simus has like a pretty strong response to that. Next thing I'll be talking about, I just want to highlight one of the promising genes we have is Win10A, which has been known to affect teeth development. And that's actually probably the biggest phenotypic difference that we see. Just comparing Simus and Blink and Beltrani, just like much smaller teeth and more teeth. And so if we just zoom in on like what's going on at that level, you can see that there's a big peak of like highly fixed SNPs all along the gene, but mostly in regulatory regions. And so we wanted to do, this is just like a very preliminary experiment. Obviously, we actually got CRISPR working in the lab, and we're trying to like do like a more better knockdown, but we just wanted to first see if like inhibiting the Wnt pathway during teeth development is a promising thing. And so this was led by undergrad Fifi. And so we took the same clutch of like IVF pupfish, we put them in different controls, so a Wnt inhibitor or a control, and then let them grow out a bit, and then looked at teeth morphology to see if just inhibiting that Wnt pathway during when we would think teeth development's happening had an influence. And we did see a decrease in teeth width. And so this is like the most basic way that we can try to get at this, but we're working on doing some more things to be more specific to Wnt10A and repeating this experiment. And so just to summarize this thing, we found that there's very different sources of adaptive genetic variation between these radiations, but we still see some similarities. So once again, Simus and the most ecologically distinct, they tend to cluster out even farther away. And so we do see that there's sort of this variable progress towards speciation, if you will. We also see similar temporal stages. So regulatory first and non-synonymous last happening in both these radiations. And I think the relative role of pre- and post-mating sexual selection might differ, but that's something I'm really interested in currently exploring. And so just to summarize, I first looked at how social environment and temperature is influencing reproductive interactions in a rat species, and found that social environment and tactic is influencing the timing of mating. So you have this interesting interaction going on. When looking at how temperature might be influencing reproductive, we see that temperatures like relevant to climate change might be undermining some of these interactions that are occurring at the gametic level. Looking at post-mating sexual selection in terms of maintaining reproductive isolation, under quite a wide range of scenarios, you can get this codivergence, sort of Scherian-esque coevolution, but it's effective at helping maintain reproductive isolation under limited scenarios alone, but with ecological divergence actually under quite a bit of scenarios. We see that different sources of genetic variation can contribute to these different adaptive radiations, but we do see a similar pattern in terms of thinking of the temporal stages of selection. And with that, I'd really like to acknowledge everyone that's made this work possible. So my current lab, Martin Lab, the Lonzo Lab, some of my co-authors, as well as the Chichikanab team, that a lot of the Chichikanab work wouldn't have been possible without. And with that, I'd be happy to take any questions. Yeah.
Could you explain again why at high levels of multiple matings, you end up with less importance for reproductive choice?
Yeah, so this is actually just all with mating. So it's nothing to do, I should have clarified that. Let me see if I have something. Okay, I'll just go back to the graph. So yeah, so let me talk with the extremes that might make it more intuitive. So at the beginning, there's just completely random fertilization success. It's just dependent on the proportion of males from population B. If you think of like a favorable mating as like a population B male matching with a population B female, at the other extreme, if females are able to sample the whole population, then they're going to be always matching and having fertilization with the other males from population B, the other migrants that are also inside.
So is the assumption that does the number of sperm not really matter? It's as long as there's preferred sperm, or can it get like swapped out? Because that's kind of what I was expecting.
Yeah, so like a swapping effect. And so you could, like, theoretically that could happen. Which it would like shift it a little bit for sure. Like this is just like a simplified version, and it's actually not quite lined up with this. It actually is like a little right shifted, I guess. If you were stronger selection is needed, is better. So it does shift the calculation a little bit, but it still has there's still that effect happening.
Okay.
Yeah.
what you have to do to collect the ovarian fluid, and then related to that, if you know that like naturally secreted fluid, if it's all from ovarian or if it's being progressively added to by different parts of the reproductive tract, and how your experiment affects that.
Yeah, it's a great question. So one of the, actually, this is pretty, the diagram's actually pretty accurate. So like the way we collect gametes from fish usually is you just like sort of squeeze the abdomen and they'll just release gametes, as well as like the fluid is literally just like it's sort of a viscous solution that's like around the eggs. And so that's how we collect it, which is pretty standard. In terms of what actually is ovarian fluid, I think that is a great question that in some fish, there actually isn't, like, the reproductive system is actually not very complicated. It's actually splenic fluid, sort of, is interchangeable with ovarian fluid. But that depends on the system. Yeah.
That's just sorry, a question. So you looked at how the different temperatures affect the interaction between the ovarian fluid and the sperm. Does the different temperature affect how the female produces ovarian fluid? Like, would the ovarian fluid itself potentially change with higher temperatures?
Yeah,
and like
so
fish stress?
that's something I really want to do. Maybe not with this system, but in other systems that's more tractable. It'd be really interesting to sort of try to tease apart effects of like immediate versus longer term effects, like the actual production itself. But yeah, it'd be really interesting to explore. Yeah, so I guess this experiment's more replicate, more of like a heat wave coming in, and where there would be less of like a acclimation effect.
Yeah, I was curious also on the RAS projects about whether you had kind of like biological or biochemical, whatever you want to call it, cellular intuition for the interaction. Like, so when you have like more sperm, but they're less fast sperm, then I can picture like some sort of trade-off happening in terms of like investment in, I don't know, mitochondria or who knows what, some sort of like cellular fuel or machinery that's needed. But then when you have this interactive effects, how do you parse that in terms of what's actually going on inside the sperm?
I think is going on, and actually keep saying I want to study, but this is actually something I want to do, is actually do an ATP concentration assay. So sort of simplify things, but sneaker sperm actually live longer than nesting male sperm. And so what I think may be going on is that, you know, temperature is increases thermal demand, and sneakers, because they have that energy storage, they're actually able to like keep up with that thermal demand. And so that's why there's not this effect of like teetering off, versus nesting males are pretty much expended at the warmer temperatures. And so I think that might be going on in terms of that. And actually, I did a review looking at differences between alternative reproductive tactics and like these different traits. And actually, there's a meta-analysis that came out recently that found essentially the same thing, that like the only consistent difference is in ATP concentration. Sneaker males tend to have, their sperm tends to be higher in ATP. In terms of what's going on with the ovarian fluid aspect, I think a lot of that could be viscosity related. So viscosity has a huge influence on these gametic interactions, and then that is also highly temperature dependent. I don't think we're really seeing denaturing of proteins at these changes of temperature, but you could have different, there's also like all sorts of enzyme interactions that could be definitely temperature dependent in this scale, but yeah. Yeah.
Do you have thoughts for why it would be more optimal for female fitness to favor the nester male sperm, and then putting that in the story with the climate change? Do you think the climate change would only be like frustrating females' ability to affect their preference, or you think it could also change what outcomes optimal?
Yeah, so I think one of the really interesting things is thinking of like the nesting males. So the nesting males, they actually go through like 3 or 4 nest cycles a season, and they'll abandon nests if they're not getting enough, if they're not like having enough success. So there could be something where if there's no longer ovarian fluid helping with that, that might influence selection acting on parental care in the system. That's like way more long-term thinking of how that works. As Julia was mentioning, there could be, you know, some sort of, there's a lot of variation too, as you can see. And so, for that. What was the first part of your question? I might have missed that.
First part was, why is it optimal for females to
Okay, yeah.
favor nesting?
Yeah, and so it's most like, I mean, it's probably genetic and environment, but what we do, the data we do suggest is that there's this like growth pathway. And so likely nesting males are higher, could be of higher quality. The other thing that's more direct benefits is that the nesting males are providing all the parental care. So there is like cooperation for that.
But can nesting males like discriminate and take better care of their own genetic offspring in a clutch?
It would be hard because like, I mean, it's possible, but sort of like there's a lot of females laying eggs all within like a very small pen. And the main parental care is like territory defense and then like fanning. It's like not sure how much control there is in like fanning. So yeah, I'm doing this motion, but yeah, that's what they do. They like sort of go around the nest and like fan their fins. And so I don't know how much fine control that affects on like the nest location, but yeah, that's a great question.
Well, if you have more questions, want to talk to Matt more about this awesome research, fierce, done them soonish.
Thanks everyone for attending.